Think of a koala. The main food source of koalas are eucalyptus leaves, but you might not know that the exact species of eucalyptus they eat can vary between different populations, the reason for this being in the gut microbiome. Different populations have evolved unique microbial communities that allow them to adapt to feeding in their environments. Therefore, eating food sources in unfamiliar environments may make them more vulnerable to toxicity. When you ingest antibiotics, the microbes in your gut can have an extraordinary range of functions that could influence their effect on you, such as detoxification or altering the efficacy of drugs. But the problem with this is that these effects are personal. If you look around, your gut microbiota is likely to be significantly different from anyone sitting around you. This leads to the question of how you can address this complexity when predicting safety of xenobiotics in humans. But that's the thing. Safety is not usually assessed in humans. Toxicity data is still largely gained from experiments using mouse models or other animal models, and these differences are even greater as humans share less than 10% of their gut bacterial species with mice or rats. Further examples of functional microbiota differences can be seen across the animal kingdom. Some populations of wood rats can happily feed on creosote bushes as part of their normal diet, but this can be fatal in other populations. So if we can look at all of these examples of where the microbiota can play such a significant role in determining an animal's tolerance to toxins, then how can we ignore this factor when using animal models to predict safety in humans? To study this, I have chosen a set of compounds where it's known that their toxicity is highly dependent on the gut microbiome. Pyrrolizidine alkaloids are produced by around 3% of flowering plant species worldwide and are common contaminants of food items that I'm sure we all eat regularly, such as teas, herbs, and honeys. There is this relatively harmless oxidized form of them, but there is also this reduced form that can go on to cause toxicity in the liver, potentially leading to more serious complications such as cancer. You can think of the toxic potential of these compounds like a scale, in which the more they tip in favor of the reduced form, the more toxic they are. Crucially, though, it is the gut microbiome that controls this balancing act, as many gut microbes can transform the oxidized form into the more harmful reduced form. The main aim of my thesis is to uncover the mechanistic basis of this transformation reaction in order to predict the toxicity produced by different microbial communities. This will help me to simulate the impact that interspecies microbiota differences can have on xenobiotic toxicity and to hopefully help answer the question, can gut microbiome differences really be reliably ignored in using safety assessment? Thank you.